Yeah, our first speaker is Rafael Lazar. He just finished his master's degree at the Justus Liebig University Gießen, where there's a lot of visual perception research going on, by the way. And yeah, he will start soon a PhD at the Center for Chronobiology at the University of Basel in Switzerland. Yep, so without further ado, please, Rafael, stage is yours.
Thank you for the introduction. I'm just gonna quickly share my screen here. Start the presentation. There we go. All right. Share screen. And the right window. So
we can see it.
Yeah. All right. I just want to start by thanking my team first. Manuel Spitschan, who is in the panel here, is actually co-authoring this research. And I want to thank our team in the Center for Chronobiology in Basel, as well as our funders who made this research possible. So I want to start by talking about pupil in general. The pupil has been a very special outcome variable used in different research. And it's so special because it's so versatile. So it has been used for risk assessment in neurological diseases and injury, as well as for detection in emotional events and sensory events. And there's also cognitive processes that can be seen by a dilation of the pupil. But most critically, it's important for visual functioning. So there's effects of accommodation on pupil size, but the most pronounced effect is its effect in response to light. And it regulates the light that hits our retina. So I'm pretty sure you're familiar with this effect from your own eyes. And that is, in a broader sense, you can think of the pupil like a bouncer to a club, basically. So when light is scarce, then we have as much light getting in there as possible. So the pupil is dilated. But once the light gets brighter and we have plenty of light, then the pupil constricts and only lets in as much as needed for high quality vision. This is also demonstrated in this video here, which I'm still amazed by actually how different the pupil can look. And on top you have the dark adapted pupil, which is fully dilated and huge in comparison to the bright light adapted pupil on the bottom row. And apart from the fascinating fact how different the pupil of one individual can look, there's these effects of inter-individual differences. So when you compare, for instance, the eyes of these two individuals, you can see that the fully dilated pupils are very different. This has also been looked at in the research of Watson and Yellett, who had a very meta-analytic approach to looking at pupil size. And then they derived a unified model from looking at 8 extant studies and computing or estimating pupil size from this data. What they used for quantifying light is the measure luminance, which is also very known for an accurate measurement or accurate estimate, that is, for brightness perception. It is basically computed by looking at the sensitivity curves, or taking the sensitivity curves of L and M cones. So let's talk about the photoreceptors real quick. You're probably very familiar. We have the L, M, and S cones, who are necessary for daylight vision or color vision, in fact. And then we have the rods, which are only used in very dim light conditions. So they saturate under very bright light, and they are necessary for us, for our rudimentary vision in very dark environments. But apart from these visual photoreceptors, there's also this more recently discovered melanopic photopigment, which is expressed by the intrinsically photosensitive retinal ganglion cells. This is very interesting because these are not necessarily used in vision, but they are used in non-visual effects. For instance, in the inhibition process caused by light, for inhibition of melatonin, I meant, caused by light. And apart from all these photoreceptors having different sensitivity curves, which do overlap though, they have differences in their temporal response to light. So melanopsin response of the ipRGC specifically has been shown to be a very slow response. So it has a long latency along with a sustained response, and it can be called very sluggish, in fact. So what is it about pupil size? How do these photoreceptors contribute? Well, all of them actually contribute, but the when and how is critical here. So when we look at photopic steady state light conditions, which we are arguably surrounded by most of the time, like now in our wakefulness, then we can see that melanopic weighted measures. So melanopsin is actually a better representation of our pupil size, shown in this graph here, than luminous measurements. So luminous would be more accurate for brightness perception, for instance. And these are monochromatic lights here, by the way. And then McDougall and Gamlin have shown that the cone response that influences pupil size is only really meaningful in the first few seconds after a light stimulus onset. And after some seconds, the melanopsin signal is the most dominant one to contribute to pupil size. We actually have quite a lot of research on the pupil. From the 20s to now, you can find a huge amount of data, but this is restricted to almost every study is restricted to limited laboratory settings. So most of them not necessarily like this old school kind of pupillography, but most of them use static setups, and only very few of them look at outdoor light, for instance, but these don't really have dynamic outdoor lighting as well. They mostly stay confined to one specific location. And furthermore, they don't use melanopsin weighted measures, but more the LNM cone weighted ones. So the question basically still remains, what does the pupillary light response look like in a complex multivariate world? So the world that we actually move and live in. And to tackle this question, we devised a new ambulatory measurement setup. So we used a spectroradiometer and put it on the forehead, adjustable in angle. And we combined it with an eye tracker from Pupil Labs. This was coordinated by a Raspberry Pi computer that made both of those devices take 10-second simultaneous samples. And curiously, we were able to move around with these measurement devices as much as we wanted to. So we were able to measure pupil size along with melanopic irradiance, or in fact, any other cone or whatever weighted light. We get this irradiance measurement along with pupil size. And as you probably can imagine, there are different lights that lead to different pupil sizes that we can compute from these still images that we took. And we measured in 7 healthy young participants aged 20 to 30. And we were able to move around freely, like I said, in completely uncontrolled conditions. And I think the most important thing is we still were able to retain 65% of our data, which was quite high, because you have to imagine when looking around, your eyes move, the bright might hit a certain spot and reflect, and we still had 65% of usable data. So these observation pairs of pupil size and spectral irradiance. The 2 protocols in which we measured these with these 7 participants, they were a little different regarding their light intensity distribution. So you can see this on the left, and on the right, you can see how closely related the photopic illuminance, so the lux, and the melanopic irradiance measurements are. And they are really closely related, as shown on the diagonal. And these outliers that you can see here are just the result of our control conditions in the lab, where we had high contrast, for instance, this blue light, which would stimulate the melanopsin much more. So the real world data is actually very highly correlated. So let's have a look at our results. We can see in this graph, we can see on the x-axis, we can see the correlations. So the higher the bars are, the higher the rank correlation is. And let's have a look at the simultaneous samples. So on the x-axis, we have different melanopic radiance samples, and these are time shifted. So this would be the correlation of the simultaneous sample of pupil size and melanopic radiance. And then when we go back by 10 seconds, looking at the light measurement 10 seconds before pupil size, then we can see the correlations start to rise. So we have a stronger correlation in the 40 seconds before the pupil size measurement, when we measure the light there, than with the simultaneous ones, which would actually be consistent with the idea of melanopsin being a sluggish signaling photoreceptor. So conservatively, what we did here is we averaged the 60-second window of light measurements and predicted this one pupil size measurement from it. And we can plot this in individual dose response curves. So this would be one individual having the data there. And on the x-axis, we have this averaged melanopic radiance in the last 60 seconds before pupil size. And then we have the pupil diameter on the y-axis. And as you can see, there's very stable prediction in the bright light and a more fuzzy prediction in the dim to medium light, which is consistent across participants, and we have high rank correlations all the way. So to conclude, we devised a novel paradigm for predicting pupil size in completely uncontrolled conditions while retaining 65% of the data. Our pupil size is strongly associated with preceding than with simultaneous samples, in line with the melanopsin signaling shown before. And apart from more research, hopefully being conducted with this setup, are, for example, assessing retinal injury risk, predicting circadian effects, or estimating our visual performance and light sensitivity. So thank you for your attention, and I'm looking forward to the questions.
Yeah, thank you very much for the presentation. Yeah, we actually have one question by Megan Vaughan. So she first says, great presentation and great idea. So what percentage of data were you expecting to retain before you began collecting? And how do you think you could improve this?
That's a good question. We were looking to, actually we had in mind to have at least a half of the data there. So I think most of the time, the problem was with very bright light conditions, we had reflections of light on the eye and a lot of eye movement. So one thing is you can talk to your participants and tell them not to move their eyes as much. And furthermore, you can look for light that is not extremely, like when you have sunlight shining directly on the light, there's less data retention. But in general, I think 50% was the one thing we were looking for, and 65% is a very good value already. So I think this is a trade-off that we have to have when moving around.
Mm-hmm. Thanks. We have another question, but I think